Okay, so we're going to continue going through the different characteristics of life. So last time we left off on the cell transport mechanisms, and now we're going to move on to how does a cell reproduce, grow, and develop. And in order to understand this conversation, we need to go back to the basics of what genetic material is. I'm sure everyone has heard of DNA before. Maybe you've heard of RNA before too, so we're just going to write some characteristics of each one of these down. So DNA stands for deoxyribonucleic acid. Deoxy, which means it's missing an oxygen, ribonucleic acid, while RNA is just ribonucleic acid. DNA is typically found inside of the nucleus. Of eukaryotic organisms. RNA can be found anywhere in the cell. It can be found in the nucleus, outside of the nucleus, floating freely in the cytosol, in multiple locations in the cell. DNA stores all of the instructions for making an individual. While RNA is primarily responsible for protein production. DNA, we have our genomic DNA, which is the big bulk of our DNA that's stored inside the nucleus of eukaryotic cells. And we also have mitochondrial DNA, which is found inside of the mitochondria. There are certain diseases that have been linked to mitochondrial DNA, and that's the only reason why I'm mentioning it here, because you may be familiar with mitochondrial DNA. Alzheimer's is one of those diseases that has an association with mitochondrial DNA. For the types of RNA, we have transfer RNA, we have messenger RNA, and we have ribosomal RNA. Those we are going to, or we may touch on a little bit more once we talk about the process of creating a protein and the central dogma. So we talked about some of the major differences now between DNA and RNA. DNA is going to be typically double-stranded, while RNA is typically single-stranded. They each use different bases. So bases are going to be the components of the DNA that give the DNA its sequence, and those bases are going to be read like instructions in order to make the protein. So the bases found in DNA are going to be adenine, guanine, cytosine, and thymine, while the bases that are found in RNA are going to be adenine, guanine, cytosine, and uracil. So they're all the same except the T is replaced by a U in RNA. And that list looks like a pretty good list for the purpose of this course. So take note of this list and make sure you review it so that you're ready to answer some questions about this on the module test. So now I'm going to go through the central dogma and talk a little bit about how proteins are made. The central dogma has to do with the transfer of information from the nucleus, the DNA in the nucleus, to the ribosome in order to make a protein. So I'm going to go back to this slide and let me just copy the picture of the cell real quick. And I'm going to add this picture here just so I can reference it more easily. So inside of the nucleus, we have DNA. DNA is double-stranded. So there's 2 strands and it's really long. Now what we want to do with this DNA is we actually want to make a copy of a certain part of it. DNA is made up of these segments called genes. And genes code for proteins. Genes can be read and turn into proteins. So think about DNA like it's a giant cookbook. And this giant cookbook contains every recipe for everything that you would ever want to make. A gene is just one recipe within the giant cookbook. So DNA is the whole cookbook. A gene is just one recipe. Now imagine the nucleus is a cupboard inside of your kitchen. And you've got this giant recipe book, or this DNA, that's found inside that cupboard. And this giant cookbook is super heavy, so you can't just carry the cookbook over to the kitchen or the rough endoplasmic reticulum in order to make whatever you want to make. You have to actually make a copy of the recipe from the book and take that copy over to the ribosome to be read so that it can be turned into what you're trying to make. So going along with this analogy, insert a text box here. Remember, the DNA is like giant cookbook. The gene is going to be the recipe. The protein that we're trying to make we can just call it a cake or whatever you think the recipe encodes for. So we're going to just keep along with this analogy as we go through here. So now we're talking about the fact that we can't just take the giant cookbook out of our cabinet. We have to make a copy of the recipe. And that's where the central dogma comes into play. Transcription. Transcription is the process of making a copy of a gene. mRNA, messenger RNA, is going to be a copy of the gene that we're creating. So when we make that copy of our DNA, we're just going to copy a particular section. So if we say like our gene is right here, we're just going to open up the DNA, we're going to copy one strand of the DNA, and we're just going to copy it. And instead of copying it exactly, it's going to be containing the base uracil, and there's some other details here that are kind of above and beyond the scope of this course. But if you were to take a biology majors class, you would definitely learn all of the details of how this is copied. So all we're doing in transcription is we're making an mRNA copy of the gene. So when we make that copy, we're literally just writing down a copy of the recipe from our recipe book. Now we're going to take that piece of paper and take it to the kitchen. The kitchen in this case is a ribosome on the rough endoplasmic reticulum. Now inside the kitchen, we are going to be reading the instructions from that recipe, and then we're going to be incorporating those instructions into, or turning it into ingredients. And that's what's happening during the process of translation. It's when a ribosome reads the messenger RNA and turns it into a sequence of amino acids. So incorporates So the ribosome is going to read the messenger RNA and incorporates a sequence of amino acids. And amino acids are just the building blocks of the protein. And it incorporates those into the growing protein. So basically, once we get to the ribosome, it's going to read what was written here on this messenger RNA. And it's going to say, okay, we need to add 1 egg, and it's going to add that egg. We're going to say, okay, we need a cup of flour. It's going to add that cup of flour. In reality, it's saying, okay, we need the amino acid methionine, and it's going to add methionine. We need the amino acid phenylalanine, and it's going to add phenylalanine. So it's reading the messenger RNA, and it's incorporating a particular amino acid based on what was written in that DNA, in that copy of the DNA. So when we talk about the central dogma, what's key is transcription and translation. But it doesn't end there. So now that we've actually created the protein, what's going to happen next? So the protein was created in this rough endoplasmic reticulum. The ribosome created it. Now it's inside here. It's going to actually travel to the Golgi apparatus, and it's going to be packaged and modified. Sometimes it's activated so that it can be functional, and then it's going to be transported to the cell surface to be released. All of those details of how this process happens, they're all a component of the central dogma, or the process of creating a protein, and that's definitely something that you would learn about further if you took an actual true biology course. But for the scope of this course, I think just knowing the basic definitions of transcription and translation and having an underlying idea of what a gene is sufficient. So moving on to how does a cell reproduce, grow, and develop? Well, we just talked about how it grows. It can grow by making proteins, and those proteins are made based on the instructions in the DNA that were incorporated in the DNA. So it goes through the process of transcription and translation, and then you have proteins, and proteins allow the cell to grow bigger. How does a cell reproduce and develop? So a cell develops, that just means that it's aging and taking on some additional specialized functions, and that can happen through transcription and translation as well. But how does a cell make a copy of itself? The very first step of making a copy of itself is it has to grow bigger, and then the genetic material has to be replicated. If we don't make a copy of the genetic material, then we're not going to have identical cells when they split in half. So you start with a cell. Let's insert a shape. So we're not going to fill this. No fill. So we have a cell here. This is our typical cell. Here we go, our cell. The first thing that the cell has to do in order to divide is the cell's going to have to grow bigger. It's going to have to be twice as big, so that when the cell does divide, both of the cells are of pretty much equal size. And inside of the cell, we have some genetic material. So we have some DNA inside of the nucleus. The DNA has to be copied so that when this thing splits in half, each daughter cell is what we call them. Each cell has the same quantity of genetic material. So that's all we're saying about DNA replication. DNA replication is making a copy of all of the DNA inside of the cell. So DNA is double-stranded. How we make a copy is we actually open up the two strands, and then we create new strands on the insides of those open strands. So now we have two strands of DNA. So DNA replication is just making a copy of the DNA inside of the cell. Now that we have two copies, now we can split the cell in half. One cell gets one copy, the other cell gets the other copy, and those cells are then identical. Why does a cell have to make copies of itself? Well, if you think about it, let's say you scrape your knee. All of those dead skin cells have to be replaced. So you're going to have to copy the living skin cells to fill in the gap with living skin cells again so that you don't have that damage. Cells are also going to reproduce to allow an organism to grow taller. So if you think about a child that's going to be growing taller, the bone cells are going to have to divide to create more bone cells, which can create more bone matrix and allow that bone to continuously grow until that person reaches about age 25, where they're going to completely stop growing. So in order for an organism to get bigger, or a multicellular organism to get bigger, or an organism to be able to replace dead cells or have more of itself, we need to go through the process of cellular reproduction. So I'm going to talk about the cell cycle for the eukaryotic cells. The cell cycle is talking about the life cycle of a cell and how a cell would create a new generation of itself. So we're going to talk about each individual component of the cell cycle, and you should take notes about what happens during each one of these phases. The cell cycle starts with G1. G1 is the first growth phase. If you remember on the previous slide, I said that the cell has to grow bigger so that when it splits in half, each of the daughter cells are still of about equal size, or about the same size as the original parent cell. So G1 is just a phase of initial growth. You can see this kind of going off on the side here where it's G0. Some cells don't divide. So inside the human body, an example of a cell that doesn't divide is a neuron. You're born with all the neurons that you have. If a neuron dies, it's gone forever. We don't make more neurons. We don't make more copies of neurons. Neurons do not divide. So they slip into this phase called G0, and G0 are for non-dividing cells only. After G1, most of the other cells in our body are going to then go through the S phase, and S is for synthesis. Synthesis is talking about DNA replication. So in the S phase is when our DNA is replicated. So we're going to make 2 copies of that DNA so that when the cell divides in half, each cell can get a copy of the DNA. Then we have G2. G2 is a second growth phase that prepares us for cell division, or prepares the cell, for cell division. All of those, so G1, S, and G2, are all part of the cell cycle phase called interphase. And interphase is when the cell is not dividing. It's getting ready to divide in the case of most of our cells, but it's not dividing. So we have G1, where it grows really big, S, DNA is replicated, and G2, it finishes up its growth to prepare for the M, and M stands for mitosis. So on the next slide, we see the phases of mitosis. So cells don't just break in half. They go through a very structured and regulated process that allows for their division. And there are 4 phases of mitosis. Let me insert a text box so that it looks better than my handwriting. So the first phase is prophase. In prophase, the nuclear envelope disappears and the chromosomes condense. So these little segments of DNA inside of our nucleus, they condense so that they can be easily separated during the next phase. The next phase is metaphase. During metaphase, the chromosomes are going to pair up and line up along the center of the cell. Now you can see the centrioles that we talked about in the organelles, and you can see these spindles forming here. All of those details are above and beyond the scope of this course. All you need to know is basically the 4 phases and what's really happening in general in these phases. So don't stress yourself too much about learning all of the details. This is just a brief overview of what's going to happen in mitosis. So metaphase, the chromosomes are going to align in the center of the cell. After metaphase, you're going to have anaphase, where the chromosomes are going to split and migrate toward opposite ends of the cell. Now, why are they splitting? Well, in the S phase of interphase, we made a copy of all of our genetic material. During anaphase, that's when we actually say, okay, this one copy goes this way, the other copy goes to the other cell. And we divide those copies equally amongst both of the cells. The last phase of mitosis is telophase. And in telophase, that's when we have new nuclear envelopes form around the separated piles of chromosomes. So new nuclear envelopes form. Now, if you can see this, these cells are still attached to each other. So mitosis just divides the nucleus into two equal nuclei, and it starts to pinch together and separate the cells a little bit. But following the M phase mitosis, we have one more phase called cytokinesis. Cytokinesis is the separating of the cytoplasms of the cell. So that finishes the division of those cells into two separate cells. So, how does a cell grow? Well, it grows bigger through the process of transcription and translation. Transcription, we make a copy of the recipe for the proteins that we need in order to make the cell grow. Translation, we actually read that recipe and turn it into a protein. We're going to incorporate certain building blocks or amino acids or ingredients in order to make our finalized product. And then we can use that product to help the cell grow. How does it develop? In a very similar way. It's going to take on specialized functions based on the process of producing new proteins. How does a cell reproduce? Well, it goes through the cell cycle. In G1, it's going to grow really big so that when the cell splits in half, we still have equal-sized cells. In the S phase, we're going to replicate our genetic material so that when the cell does split in half, each cell has equal amounts of DNA. G2 is the final preparation for mitosis. So all of that happens in interphase. Once we enter into mitosis, the nuclear envelope disappears and chromosomes condense. So our chromatin condenses down into chromosomes. Then we have metaphase, where our chromosomes align in the center of the cell. So each one of these is one copy of the replicated DNA. Then in anaphase, those copies are going to separate and migrate towards opposite ends of the cell, so that each cell is going to have the same identical kind of genetic material in it. Then we enter into telophase. And telophase, new nuclear envelopes form around the separated chromosomes. Following that, we have cytokinesis, which is the splitting of the cytoplasm. So that's what finishes up dividing the cells into two separate cells. And cytokinesis happens after mitosis. So in general, that's how cells are going to reproduce, grow, and develop. So moving on to the next topic here, how does a cell process energy? Well, all organisms on Earth are going to interact in a particular way to exchange energy. And that's what life is dependent on, is an exchange of energy. Energy comes from the sun. If we didn't have the sun, life would cease to exist. So our initial energy comes from the sun, and it strikes down on these producers. Producers use energy in order to produce their own food. Other organisms, though, can also eat that food by eating the plant. So our producers are going to provide or produce food for all of the other organisms on Earth. The consumers obtain energy by eating other organisms. So they're either going to eat other consumers or they're going to eat the producers. These decomposers are going to consume both producers and consumers after they're dead. So it's going to get nutrients from dead organisms and organic wastes. Now there's a particular process that cells go through in order to create energy. You just need to know the names of the three parts. You don't have to know all of the details for this. So, how does a cell process energy? Well, the cell goes through something called cellular respiration. And let me just type that in here. Other processes include fermentation or photosynthesis. This diagram shows cellular respiration. The three parts of cellular respiration are glycolysis, the Krebs cycle, and the electron transport chain. So those are the three components. Glycolysis happens in the cytosol of the cell, while the Krebs cycle and electron transport chain are going to happen inside the mitochondria. Each one of these is regulated by enzymes, and each step within here has a lot of different pathways that can happen, and that's where we get into more of the biochemistry metabolism type discussion. So we can talk about how those enzymes have an impact on all of the different components of these cycles and processes in the field of biochemistry. Fermentation is when we produce energy, but we also produce either an acid or an alcohol as a waste product. So people, when we go through the process of fermentation, it's because our muscles become oxygen deprived and we switch to fermentation or anaerobic respiration. And in that process, we end up producing lactic acid, which is why you kind of feel your muscles burn after an intense workout. But fermentation happens all the time with microorganisms. We can produce different types of acids, but also alcohol. So when yeast, certain types of yeast ferment, they produce alcohol. And that's where we get our alcoholic beverages from, is through the process of fermentation. Photosynthesis is the process of using sunlight in order to make a food source. After an organism makes their own food, then they have to take it through cellular respiration to get energy from that food. So that's, in a nutshell, how energy is processed. Again, if this were a majors level class or a biochemistry class, you would definitely be learning about all of the ins and outs of these processes. But for now, we're just doing a basic overview. The last topic that we're going to discuss is evolution. So how does a cell evolve? Evolution is one of the necessary life functions, and an individual organism cannot evolve. Only a group or a population of organisms can evolve. So evolution occurs in a population when allele frequencies change from one generation to the next. Allele frequencies. We talked about genes previously. We say genes are the recipes to create proteins. Alleles are forms of the gene. So you can have a gene for eye color. And let's say like a fly can either have red eyes or brown eyes. Allele is the color for that particular gene. So there's a gene that controls eye color, but the alleles would be either the red allele or the brown allele. And when we talk about alleles, we start to get into genetics a little bit. So if you're interested in learning about heredity, genetics is definitely the field that you would be looking into. When we talk about allele frequencies, we're talking about the number of a certain type of form of the gene in a population. So if one generation had 50 red eyes and 50 brown eyes, and the next generation had 10 red eyes and 90 brown eyes, we would see allele frequencies change from one generation to the next. And that is the technical definition of evolution. So this is definitely something that we can see happen even within our lifetime. We can see evolution occurring. And using that kind of logic there, we can see how evolution is defined. So now we go into survival of the fittest. Survival of the fittest means that the best kind of more capable organisms are going to be able to live. And those living organisms are going to be able to produce offspring. And those offsprings are going to have the characteristics of their parents. So the alleles of their parents are going to be found in the offspring, and those are going to be the traits that are going to survive through the next generation. So we see this kind of go through here. There's genetic variation. So naturally, through mutations, through random chance, through random assortment, we have a variation in every population on Earth. There's limited resources, so every habitat contains a limit to how many resources are present or supplies that are required for survival. Then we have an overproduction of offspring. So we produce way more offspring than can actually survive in that environment. So let's think about rabbits. I mean, rabbits reproduce like crazy. So we have genetic variations. Some rabbits have stronger back legs, some have bigger ears, some are a little bit fatter, some are a little bit heavier. There's limited resources, so there's only so much grass and leaves and plant material that the rabbits can eat in a particular area. And rabbits are going to keep producing offspring. So one rabbit just keeps multiplying and multiplying, and now you've got a huge amount of organisms that are trying to survive with this limited amount of resources. So it turns into a struggle for existence. Some organisms are going to have to fight for those food sources. Because they're fighting, some of them may end up losing and getting injured to the fights and end up dying, while others may not be strong enough to fight and they may end up dying of starvation. While the ones that are strong and healthy and that won the fight are going to be able to reproduce. So there's unequal reproductive success. The stronger ones are going to be able to live, and they're going to be able to have the next generation of babies. And then you have descent with modification. Because this happened, now we've got stronger bunnies that are reproducing, and those stronger bunnies are going to pass on those strong alleles to their babies. And those babies with the strong alleles are going to pass on those strong alleles to their babies. And eventually, all of the weak bunnies are going to disappear, and we're just going to have strong bunnies out in the population. Now, this is just an example. There's different things that confounding factors and stuff, predators and all that type of stuff, cars running them over. But this is an example of how evolution can occur, and survival of the fittest plays a role in how organisms are going to evolve. Now remember, evolution cannot happen within one organism. Evolution happens within a population of organisms. The last example that I'm going to talk about is antibiotic-resistant bacteria. So, when you get a bacterial infection, you're going to go to the doctor. They may or may not culture it. They may just look at it and prescribe you an antibiotic. Well, bacteria typically within a culture of bacteria, within an environment of bacteria, there's usually one that is resistant. How did it get resistant? It just is. There's mutations that happen, there's random events that happen, and randomly one of those within the population is just going to be resistant already to the drug. It's not like we gave it a drug and it developed resistance. It already existed in the population. So let's say the doctor gives you an antibiotic, and the antibiotic is going to kill most of the bacteria, but this one doesn't die. Normally, that's not a problem. So under normal circumstances, if you take your full dose of antibiotic that's prescribed, that full dose is going to kill all of the ones that are susceptible, leaving this one guy, and your immune system is just going to eat that one guy. But what a lot of people do is they start taking an antibiotic, and then they either forget a dose, or they're like, oh, I'm feeling better, so I'm just going to stop taking it, which means that you have a sublethal dose. Sublethal means that not all of these microorganisms are killed. So now you've got maybe 10 of them killed, and the rest of them are around. Now, when your immune system starts to respond to this, it is going to try to kill all of the microorganisms, but this one that's resistant is going to have a chance to multiply because it's hidden amongst all of the ones that are not resistant. It has safety in numbers now because it did not receive the appropriate dose that it needed to kill off the ones that could have been killed. So now what you see is you have the microorganism that's replicating like crazy in the presence of the antibiotic. So even though the antibiotic is there, that it's only able to kill the ones that are susceptible, and the one that was given an opportunity to grow is now going to grow. This process in and of itself is evolution. It's the change of allele frequencies from one generation to the next. In this generation, that resistance gene didn't exist. In this generation, one was resistant. Multiple generations later, you can see that there's a lot of resistant microorganisms, and that's typically due to human error or over prescribing antibiotics.